Plasmids are circular DNA molecules that are tagged back of independent way to the chromosome of the host cell. Of natural way, they are in the bacteria in sizes that go from 5,000 to 400,000 pairs of bases. Synthesized plasmids have been obtained many from which they happen of natural way. All the vectors of donation must contain at least 1. An original of replication to be able to have more than a copy of himself in the infected cell. 2. Two genes that confer resistance to different antibiotics, which allow the identification of the cells that carry to this vector. Plasmids can confer properties daily as resistant to antibiotics as amino acids, chloramphenicol, beta-lactamase, heavy metals, the toxic anions like acetals, arsenic, borates, and chromates. In general, these plasmids contain genes that codify proteins that they inactivate to the toxic compounds for cells or affect his transport towards the interior of the cell. For example, production of beta-lactamase, enzymes modifiers of amino acids, chloramphenicol acetyltransferase, or metabolic properties, production of bacteriocins, carbohydrate metabolism, carbon diamond metabolism compound, and nitrogen fixation. The general steps for purification of the plasmids are the following. Obtain a milliliter and a half of the culture medium it was previously grown overnight. Subsequently, to remove centrifuge culture medium, perform result suspension package. To carry out the alkaline lysis, larger particles by centrifugation or filtration membrane are removed. It may be an intermediate centrifugation or high resolution. Otherwise, it's a start.
Centrifuge the culture 20 seconds to obtain the bacteria's pellet. Perform a wash to the pellet by resuspending in 500 microliters of STA buffer and centrifuge at maximum velocity and discard the supernatant. Resuspend the pellet in 100 microliters of GTE solution, complementing with 2 milliliters of lysozyme and mix in the vortex. Add 200 microliters of alkaline SDS solution mixed by immersion 5 times. Add 150 microliters of high solution, mix in the vortex for 2 seconds and put the microtube on ice for 5 minutes. Centrifuge at maximum velocity for 5 minutes and transfer the supernatant into a new microtube. Precipitate the plasmid through adding 2 volumes of absolute ethanol. In this case, the added volume was 1 milliliter. Mix by immersion 4 times and incubate at room temperature for 5 minutes. Add 1 milliliter of ethanol, 70%, and centrifuge for 2 minutes at 4 degrees Celsius. Dissolve the supernatant using molecular biology water, and finally verify the plasmid's integrity by performing an electrophoresis.
3 isoforms are observed in the gel, which correspond to supercoiled plasmid, relaxed, circular, and linear, which have different sizes. The first to be observed are bands 4,000 base pairs and linear bands. Then you can see the circular bands relaxed and approximately 2,250 pairs, and finally the supercoiled less than 300 base pairs. When the plasmidic DNA of the bacteria is extracted, something is obtained that considered a purified product, but in this case can be seen molecule of plasmid in 3 conformations, logically different forms at what person. These 3 forms, now like supercoiled, relaxed, and linear complex length, correspond to the same molecule and have all the meso number of pair of bases, or leg of the molecule, but have different form. Reason why they occupy different and effective volume and they advance by the gel at different speed.
Ensure destruction of the plasmid and not destruction of other genetic material, it's important that after dissolve the pellet with molecular biology water, give a treatment with DNases or RNases to avoid the contamination with DNA or RNA, and also perform another phenolic extraction to eliminate the enzymes. Depending on the nature of the plasmid in the agarose gel, two bands have to be observed, one corresponding to the plasmid and the other one corresponding to degraded RNA. In agarose gel that corresponds to the practice, three bands can be observed. The first one could be a relaxed circular plasmid, the second one could be a linear plasmid or a supercoiled plasmid, and the last band could be a degraded RNA.
It's normal that in the yellow agarose of they observe three bands due to isoforms of the plasmid. The molecules of plasmidic DNA are circular but, like in the bacterial chromosome, is twisted due to the enzyme GRAS, that is one topoisomerase of type 2. In analyzed cell, all the molecules of the plasmid one stay in this supercoiled form thanks to a continuous cost of energy. Finally, in the bands observed in the gel, it is possible to be seen that if count on the three forms of the plasmid one with its corresponding sizes. The method used for the obtaining of the plasmid is denominated of alkaline lysis. It is based on the desnaturalization and selective precipitation of the DNA chromosomal in means with hydroxide of sodium and SDS. In these conditions, the plasmidic DNA mainly remains intact due to their small size and its circular and supercoiled nature. The treatment with high concentration of salt and SDS produces the precipitation of great part of proteins. The treatment with RNase eliminates the RNA contamination that can be important. The purification of the plasmidic DNA is completely done by precipitation with ethanol. It's possible to obtain plasmidic DNA with different degrees from supercoil that can be visualized after the preparation by electrophoresis and tinction with bromide of ethylia.